Hello, everyone, and welcome. Thank you for joining us for today's session, the third in our new webinar series titled Environmental Monitoring Considerations for Gene Drive Modified Mosquitoes. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Our mission is to support and advance best practices and informed decision-making for the development of genetic biocontrol technologies to improve public health. To do this, we work to identify and address key questions, provide technical advice, and strengthen capacity and information sharing. An important part of the testing pathway for new genetic vector tools will be post-release monitoring for safety and efficacy. This webinar series will highlight current vector and biodiversity monitoring efforts across Africa and introduce potential methodologies and tools that could be used to support environmental safety monitoring following release of a gene drive modified mosquitoes, which are being developed for malaria control. Sessions will take place weekly through the first week of June. Before we get started, we have a few housekeeping reminders. Please email events@fnih.org if you have any technical issues. Your video and audio will be off during the webinar. The presentation will last between 45 and 60 minutes. While the presentation is going, feel free to submit questions anytime in the chat. Questions will be addressed at the conclusion of the presentation, and all webinar recordings will be posted on the Gene Convene Virtual Institute and the Gene Convene Virtual Institute YouTube channel. Today's webinar is titled the National Ecological Observatory Network, also known as NEON, using big data to enable research and to understand changing ecosystems. And we are honored to be joined by Dr. Stephanie Parker. Dr. Parker is an aquatic ecologist with the National Ecological Observatory Network, which is operated by Battelle and based in Boulder, Colorado. Dr. Parker holds a BA from Middlebury College, an MS from the University of Maine, and earned her PhD from the University of Alabama, where her research focused on how Arctic stream food webs respond to natural disturbances. Since joining NEON in 2010, she has played a key role in shaping and maintaining a number of their aquatic sampling protocols, including those for algae, aquatic microbes, aquatic plants, microinvertebrates, and zooplankton. In addition to protocol development, she works closely with field ecologists on training and troubleshooting, partners with external contractors for sample analysis, and conducts regular quality checks across multiple aquatic data streams. Her experience with developing consensus collection protocols and analysis methods should be very informative for those who are interested in post-release monitoring of gene drive mosquitoes. Before we get started, a quick reminder. Following the presentation, I'll be monitoring a Q&A session, so please feel free to post your questions in the Zoom chat at any time. Additionally, today's session is being recorded and will be posted to the Gene Convene YouTube channel after the event. With that, I'll turn it over to our speaker. Dr. Parker, if you can go ahead and share your slides, we can get started.
Great. Thank you, Justin, and thank you all for having me today. It's a pleasure to be here. As Justin said, I am from the National Ecological Observatory Network. I'm based in Boulder, Colorado, although we have sites all over the country, and you'll see that in just a moment. There we go. Sorry, I have an extra bar over my slides here. So the National Ecological Observatory Network, or NEON, is a continental scale, long-term observatory. The plan is to be collecting data for 30 years. NEON is funded by the National Science Foundation and operated by Battelle, which is based in Columbus, Ohio. NEON enables the analysis of free and open data and makes samples available that can be used to assess the drivers and responses to environmental change. NEON also enables comparison of across data, across sites, using a standardized and reliable framework for research and experiments, and provides interoperability, so integration with other national and international network science projects, which I'll talk more about near the end of the talk. NEON is important for ecology because it provides a highly coordinated national system for monitoring a number of critical ecological and environmental properties across a wide array of spatial and temporal scales. So we have sites, field sites in Puerto Rico, Hawaii, all across the continental US and Alaska. And again, I mentioned that our plan is to collect data for 30 years. So that's a pretty long temporal scale. compared to most other ecology projects. NEON provides open, fair data, provides samples and infrastructure to enable open science. So if you're not familiar with fair data, that's data that are findable, accessible, interoperable, and reproducible, which are all really important properties if we want a multitude of other data users to come in and use our data and connect it up with other networks. NEON is a United States NSF large facility, that's how it's classified, and is a continental scale observatory that's designed to enable the understanding and forecasting of the impacts of environmental change. So across the observatory, we have 81 field sites. 47 of those are terrestrial and 34 are aquatics. And I work on the aquatic sites. We provide over 180 different data products through our open data portal. And as I mentioned before, we'll be producing 30 years of data. NEON produces this data using linked collection systems. So again, data collection, sample collection, instrumentation, excuse me, is standardized and co-located across the 81 sites. So we have several sites where we have a terrestrial site and an aquatic site so that data users could come in and pull data from both of those data streams and be able to say something about effects of aquatics on terrestrial or vice versa. The link collection systems include automated instrumentation, and we have this instrumentation, as you see over here, as towers in the terrestrial environment, and then we have sensors in the aquatic environment, buoys in lakes and rivers. We also have an airborne remote sensing system that's mounted on airplanes that fly over certain regions of the country on a sort of rotating basis from summer to summer and collect LiDAR data and a number of other remote sensing data products. And then we have observational sampling, and this is what I know the most about. And this is where our field ecologists that work out at all of the NEON sites go into the field, collect samples, collect data, maintain instrumentation, and either do some analysis at their domain support facilities or send those samples off for further analysis from expert labs. In addition to the data that is generated from these three subsystems to the 180 different data products, we also have samples that are sent to the NEON biorepository at Arizona State University. So NEON has more than 100,000 samples going to Arizona State every year. And this is a great opportunity for researchers who have questions that NEON is not answering during our regular sampling. They can go grab those samples and basically do whatever analyses they want to on them. So NEON, as I already mentioned, provides a broad range of open and curated data. We provide data from the atmosphere, so water, carbon dioxide, heat. We have micromet stations. We provide data on isotopes, turbulence in the atmosphere, storage and fluxes. We do a lot of work with organisms, populations, and communities, so we get data that can be used to assess biodiversity, productivity, pathogens, which I know a lot of people in this group are interested in, and phenology and metagenomics. We also do ecohydrology, so water quality, precipitation, discharge, geomorphology, biogeochemistry, both on land and in water, and land use and processes. And a lot of this comes from that airborne spectrometry and LiDAR data. So NEON can be used to enable ecological forecasting, which is kind of a really new place to be in, where we have standardized sampling and data coming in that can be used across these wide spatial scales and used for a long time period. So we do collect data that can be used to assess fine scale processes. So at the organismal level, if people are interested in that, we have data that can enable analysis at the regional scale, and then kind of moving up through patterns of distribution and abundance to remote sampling and point sampling, and then up into spatio-temporal modeling and forecasting. And what's really great about NEON is that we do provide data that can be used at all of these different levels. So NEON exists to provide data for other people to use. And if people aren't using it, what's the point of us being here? So we provide data at all these different levels that can be used in a multitude of different projects and research programs. So going all the way from individuals to populations to communities to ecosystems up to the landscape, you can look at energy transfer all the way up to biogeochemical cycles. As I mentioned before, NEON uses open and fair data principles. Very important for scientific data management and stewardship, especially going forward in the era of bigger data. NEON was designed from the outset to be an open data generator and provider, so there are no paywalls or anything like that. All our data are freely available, quality checked, and available to anybody who can access our data portal. And again, we strive to make the data findable, accessible, interoperable, and reusable. NEON's contributions include the data, which I've been talking about. So open data for over 180 data products, standardized co-located protocols, and collected across several different taxa, time, and space. We also collect specimens and samples. Samples that are not used up in the analysis process are sent off to be archived for the life of NEON at the Arizona State NEON Biorepository and are available for request and for research and educational use. NEON also provides, in addition to the samples and the data that we keep talking about, community and workforce development. So contributing to information and data management standards, providing interoperability with other data sets. We provide training and then support to data users because it can be pretty overwhelming to come into the NEON data portal and try to figure out what data you want to use and how you're going to use it. We also provide research infrastructure. So there's a program that's called Research Support Services at NEON, where data users can submit a proposal to either add additional field sampling, like have NEON field ecologists collect additional samples for a separate project, add sensors to our existing infrastructure, users can request additional remote sensing. We also have mobile deployment platforms, which are really cool. They're sort of like mini towers or mini instrument arrays that go in aquatic environments. And there's, again, always access to NEON experts. And we provide field site coordination along with this as well. So the NEON specimens and samples, this is a really huge undertaking. The NEON Biorepository is located at Arizona State University and is sort of a separate facility from their regular museum. We are contributing over 100,000 specimens and samples per year to that biorepository and working with other groups such as Barcode of Life, GBIF, and iDigBio. There's over 65 different types of samples that go down to the biorepository, and I pulled these numbers just last week. So right now, as of now, we have more than 800,000 samples, NEON samples at the biorepository, and that covers almost 6,000 different taxa. So samples that go to the biorepository include small mammals, fish, ground beetles, mosquitoes, ticks, zooplankton, macroinvertebrates. We also send plants, microbes, soils, there's dust samples, wet deposition, water samples, and many more. And everything over on the lower right that's in blue also includes associated genomic data. So DNA extracts exist for these as well that can be requested and used by researchers. Going back to our NEON Research Support Services, this has been a pretty popular program, and it's a really great opportunity for someone who has a grant to come in and leverage the NEON sites and the existing information that we're already providing and the existing infrastructure, and maybe take some of the burden off of them for being able to get to the field as often as they want to, or being able to establish some of that instrument and infrastructure that they would need for their project. So this is PI-driven research, where PIs have access to our observational sampling structure. So we have several projects right now where our field staff just collect an additional water sample every time they're out, come back to the lab, either filter or freeze it, and then ship it off back to the PI so they can do whatever analysis they want to with it. There's also access to sensor infrastructure. So again, adding different instrumentation than what NEON is already providing to our infrastructure. We can add packages to the airborne observation platform. And then again, there's these mobile deployment platforms like we have in the middle. That's sort of like a little mini tower. And that has been used in events like forest fires and things like that. When sort of a sudden disturbance event happens, researchers can request this platform and get a whole suite of NEON instrumentation that's being used at NEON sites so data could be compared. So by the numbers, we have a lot going on right now at NEON. We have more than 600 staff. So that's full-time, part-time, seasonal science staff. So that's quite a bit. And we have those staff distributed all over the country at all of our domain support facilities. NEON samples in 20 ecoclimatic domains. We'll talk a little bit more about that, but the country was split up into domains or ecoregions, again, from Hawaii up to Alaska, and that encompasses 24 states and 1 territory. That's Puerto Rico. We have a number of different assets. We have those airborne payloads that I talked about before, more than 16,000 sensors, which all have to get calibrated every year as well to make sure they're functioning properly. Lots of community-led and NEON-supported events every year, and 5.6 billion data points daily, which is a lot. That's a lot of data. And how do people deal with these big data sets? And especially in ecology, this is sort of an emerging field. So NEON numbers, I mean, the numbers are big. There's a lot going on, but they don't tell the whole story of how this large interdisciplinary facility functions, right? There's a lot of other stuff going on behind the scenes. I talked about instrumentation and sampling and what we do with the samples already, but there's this whole infrastructure on the back end to manage the data. And we have to have our cyber infrastructure group and people who can manage the data portal and provide that free available data to our users. So we start way back here, observational sampling and instruments and remote sensing and mobile deployments, and all of that has to go in to our publication and release system, right? We need to be able to back up the data. We also transition them from sort of raw, we call it level 0 data, to something that's slightly cleaned, slightly processed, and that's what we provide to the data users. And then there are other data products with higher levels, like up to level 4, where those are highly processed data products. So if you know anything about aquatics, stream flow and discharge is a highly calculated data product, and that is a level 4. So getting into the weeds a little bit there. We now use Google Cloud Platform to store a lot of our data. And this is how the data users access it via our data portal. So once those data are available in the data portal and accessible to users, we are starting to see more and more publications using NEON data every year. If you are interested in publishing using NEON data, please do check out, please check out the, we have citation guidelines on our website, and that's how we can find these papers once they've been published, because they've been cited in the NEON format. So as of early April, about a month ago, we had over 27,000 citations using NEON data, which is really great. And these data are pulled from NEON publications from Dimensions and GBIF specimen records. And these data are being published across a variety of different types of science. So terrestrial, aquatics, atmospheric science, and that's really exciting. You can see the numbers have been going up and up every year because we have more data available, more people hear about us. And obviously we're still pretty early in 2025, but hopefully those numbers climb up as well. So 2024 was a pretty good year for publications using NEON data. And I have to say, as a NEON scientist, it's really exciting to go to conferences. I've worked at NEON since 2010, since we were setting up the observatory. And it's really exciting to go to conferences now and just see talks about NEON data. And usually they're people I haven't met, and they've just accessed the data portal and they're doing their own thing with them. And that's really, really great. And that's what it's all about. So you might be wondering how we got here. I know that FNIH is trying to set up its own monitoring program for malaria, but how did we get this all started for NEON? Well, it's been a pretty long process. Neon was conceived in the late 90s during meetings of ecologists and biologists. So the original idea is that neon is a telescope that's trained on Earth, right? We talk a lot about the fact that we're an observatory, and when you think about observatories, you probably typically think about space observatories, obviously. There's oceanic observatories, but this is the first ecological observatory of its kind. And the scale, it was determined early on, researchers wanted this to be the entire continent, right? So this took hundreds of scientists and engineers that contributed to the initial planning and design and operations of NEON over a number of years. Since the late 90s, obviously technology and research tools have evolved rapidly. We're seeing that every year. And that includes instrumentation that evolves rapidly, the way the data portals are set up and access, where we store the data, how much data we can store, and going right down to like genetic methods. Those are changing all the time as well, and we have to keep up with the times to make NEON data relevant. So NEON is the first big ecology project. It was constructed with NSF major research equipment and facilities construction funding, and that's the funding that is typically used to build things like telescopes and space observatories. The funding had, well, there we go, and particle accelerators, that was my next point. And the goal of NEON is to improve understanding and forecasting of ecological change at continental scales. So the brief history, in 1999, there were discussions about a, quote, biodiversity research network. From 2000 to 2005, there were a series of workshops that were held to develop the ideas for NEON. To be a more comprehensive ecological observatory network and expand beyond just biodiversity, like the original thoughts, and address continental scale questions. And in 2006, that initial plan was completed. So these early workshops were held by researchers in the field of ecology from all different institutions and agencies. And that's where our data products initially got set. That's where people said, we want to look at aquatics and terrestrial and airborne and instruments. And these are the organisms or instruments or data streams we want to have in each of those categories. From 2006 to 2011, that's where the designs, the plans, the budgets were formally reviewed and revised. And that was a lot of work with NSF, with technical working groups, trying to get all of those protocols and instrument plans kind of figured out and figure out exactly what data we were going to provide to the data users. And of course, this is all with community input. We're providing data that the community wants. Because if the community doesn't use our data, again, what's the point? In 2011, the funding for NEON was approved by NSF, the National Science Board, and Congress. So this is a line item in the congressional budget. And then construction started in 2012. And so construction was building out the field sites. Each field site needs to have line power and line communications, if possible, to get keep all the sensors running and get data back to the NEON data portal. Not all sites have those things. It just wasn't possible for some of the more remote sites like in Arctic Alaska, and there were other ways to work around that, using satellites and generators and things like that. And I don't want to get into that here, but in general, construction was kind of a big deal. Permitting was hard for a lot of the sites. We had a lot of sites selected in those early days and went around to kind of do some analysis and figure out where things would be at each site, where instrumentation would be, where groundwater wells, things like that. And then sometimes, permitting would just fall through and we'd have to go find another site. And then in 2019, this was really exciting. That's when all the construction was complete. So all the sites were up and running and functional, and NEON enters what we call the operations phase. And this is the first year of that 30-year timeline, so 2019. And we do have data prior to 2019, but we consider that legacy data, so data that we're streaming in, and we learned a lot from that. We learned a lot about how the processes worked, how just little things like shipping to external facilities worked and how data could stream onto the data portal and what sort of challenges we might have with that during that time. But all those data are available, quality checked, and if it's on our data portal, it's good to use. So some of the early concepts of neon. So these are some of the artistic renderings that were done as early as 2007. But basically what I wanted to show here is that all of these, going from 2007 up to what we are doing now with neon, they've changed a little, but the big factors are still here. So we still have an airborne platform. We have aquatics in each one of these. We have forested sites, terrestrial data coming in. We have towers with instrumentation, and this is the plan all along. And so the details within that might have changed over time, but the basic structure and framework has not in terms of what we're providing to people. NEON was initially set up around these 7 grand challenge questions. 6 of the questions were originally identified by the National Research Council in a publication in 2001. And so those grand challenge questions were to look at some causes of change, so climate change, land use change, and invasive species. Invasive species was the one that was not included in the NRC report and the one that was identified and added by NEON planners, and probably a good thing it was because invasive species seem to be more and more important every year to ecosystems. And other grand challenge questions were classified as responses to change. And so that's biogeochemistry, biodiversity, ecohydrology, and infectious diseases. And you can kind of see these themes going throughout all the NEON data products if you've gone and poked around on our portal a little bit. So these 2 groups, causes of change and responses to change, were initially set up, but the idea is that the causes and the responses could switch depending on the scale and process. So for example, the vegetation structure can affect climate, and climate can affect vegetation structure. Excuse me, can't say that word today. Which is sort of an interesting way to kind of look at and classify our data and group data products together. So why is NEON different from other data streams that you might see? NEON data and resources enable users to tackle scientific questions at a scale that has not been accessible to previous generations of ecologists, which is really cool. I think of people I worked with when I was doing my dissertation or postdoc, and those were pretty small projects. You know, those projects lasted the lifespan of a grant. So a few years, maybe 5 years if you're lucky. And were confined to you know, 1 or 2 regions that people could get to because, you know, we didn't have the people to go collect all of these data and have all this instrumentation out and honestly have the permitting to be able to study all these different locations. And those are things that NEON provides. The primary purpose is to provide, again, continental scale environmental data and archive samples and infrastructure for ecological research studies. And on top of that, we want people to use the data because, again, I've said it a few times, if people don't use the data, there's no point. So we need to provide educational tools to enable people to work with our data sets and our large data sets. NEON data and samples are collected in a standardized, consistent, and comparable way. They're integrated, so you could look at data, biological data and chemical data and physical data all from the same site and date and make do some analyses on that if you wanted to. And then field collections and in situ and remote sensing are all part of this plan all along. So getting back to how we were designed, NEON was designed by the community. There was no NEON before like 2009 or 10. There wasn't like a headquarters office with scientists at it. So it was designed by the community. Initially, this started with the community selecting field sites. So NEON is organized in core field sites and gradient field sites. The core field sites were initially suggested by the community. A lot of them are sort of at that centroid of those ecoclimatic domains or ecoregions we have set up across the country. Those needed to be representative of that region. So for example, our domain 5, which is sort of the Wisconsin region for aquatics, we look at lakes up there because lakes are what the common aquatic habitat is, right? And we don't have a lot of other lakes across the Neon system, but we do there because lakes are really dominant on the landscape. So that's an example of how sites represent the ecoclimatic region. Some of the sites that were selected have already had a history of research, like long-term ecological research projects. And then we have gradient field sites, which are designed to look more at like climate, elevation, land use changes. So just to go back to the core sites, we usually think of those as more of a sort of a pristine site unaffected by humans at the moment. And then gradient field sites can have a little bit more going on at them. So in 2006, there was a request for information put out to the scientific community to propose questions. And those questions are what drove the selection of these gradient field sites, which is a pretty cool way to pick some field sites, I think. So scientists in the community were able to select these sites and select questions that could be asked. Some of them use like a cause and effect paradigm, and some are selected to answer ecological questions. So over on the right here, I have an example of our domain 8. So that's the Alabama, southern US region. And for aquatics anyway, those sites were set up along a hydrologic gradient. And that was proposed by scientists at the University of Alabama who were already working in these systems. So these sites range from Mayfield Creek, which is a small wadable stream in the Talladega National Forest. Then we have the Black Warrior River, which is a larger river that Mayfield Creek flows into, and then that flows into the Tombigbee River, which is an even larger river and goes out to the Gulf of Mexico. And so that's how those gradient field sites were selected. And there's a lot of other stories like that across the observatory. The observational sampling, instrumentation, and airborne platform, again, just like with the field sites, designed by the community. These data products and scope were set up by an early science design that was published back in 2009, I believe, where NEON scientists, along with technical working groups, and I'll get to that in a little bit, wrote the initial protocols. A lot of those protocols were based on existing sampling and historic work in the areas. I can say for myself, protocols that I wrote for aquatics, I tried to lean on USGS and EPA as much as possible to align all of those sampling methods so data could be compared across systems. And then we had to design the data products and the data and samples that were coming from them to fit within the budgets that we have that were set by NSF. We also have our cyber infrastructure group, which again, with the technical working group, so experts in the field developed and tested the data portal. And all of this happened with input, continuous input from the scientific community, and then ultimate approval by NSF before anything was operational. And then to add on to this, that's how we set it up. But now that we're in the operations phase, our sampling protocols, instrumentation, statements of work that get sent out to external contractors, we have them regularly reviewed by the scientific community, by our technical working groups, to make sure we're still asking for the things we think we're asking for, everything's clear, and everything makes sense when it gets out there to the public. So standardization is a huge piece of NEON as well. We standardize data collection and instrumentation across the entire continent, and it's really, really hard to do that. It's really daunting. I work in aquatic systems. Water washes things away. It's hard to make things stay in a stream, especially when you're having a spring flood or a big rain event or something like that. So on top of that, just getting out and sampling in the same way in a stream that's in Puerto Rico versus Arctic Alaska, like those are two different things. And how do we standardize those protocols? And even beyond that, we have streams that, like desert streams that dry out in the summer. We have streams like our high gradient mountain streams that freeze solid or have so much snowpack our ecologists can't get in there. We have wide streams, we have agricultural streams, we have this whole range. And how do we standardize? Well, it took some trial and error. We initial protocols and instrumentation were what seemed to work at other sites and other systems and in other sampling networks, LTRs and things like that. One great example is the way our sensor instrumentation is set up in streams. So all of the sensors initially were set up like this top right set of sensors where they're attached to the stream bottom, which works fine as long as the stream doesn't flood too much. If too much water velocity gets going through there, that'll wash those sensors right out. So after losing some sensors, as you do, or not losing them, but they fell out of place and weren't where they were supposed to be, our engineering team, along with the aquatic instrumentation scientists, design an overhead cable design where a cable stretches across the creek. This is kind of hard to see. This is from one of our phenocams. And there's a rod hanging down with the sensors attached to that. So when the water levels get high, those sensors can kind of float up and still collect data and not be washed out with every flood. So some trial and error, some things need to be enhanced and made better over time, but I think we're in a good place now with that sort of thing. So community requested changes begin with science, right? We have had this community input all along in the NEON process, and that doesn't stop now that we're running and that we're in the operations phase. So typically there can be some kind of user request, like sometimes we'll get a question from a scientist using our data, and it could be something like, well, I'm going to go into an in-depth example in a little bit, but it could be something like, well, why aren't you collecting this? This is a really common thing to collect, right? So that goes to the NEON subject matter expert, the NEON scientist, to think about that and like, oh, is this a good idea or not? Then that scientist works together with our technical working groups to think about the impacts of that, how important it is, how useful it would be to data users, get some costing and some proposals. That then goes to an integrated project team that's within NEON that is composed of a variety of different scientists and engineers across the NEON program to review and approve this and think about the budget and how important that is. If that is approved, that then goes up to another round of external support, and that's our Science, Technology, Education, and Advisory Committee, or our STEAC. And they can review the proposal and recommend or not. And so their recommendation actually goes to the NSF, or goes to our observatory director, and then that goes to the NSF for review and concurrence. So any changes that we make to our instrumentation or data products now, anything that's a significant change, goes through all these steps. It goes through science, technical working groups, the STEAC, the observatory director, and all the way to NSF. So there's never any places where people like me, just the scientists, are making like unilateral decisions. That doesn't happen. This is totally a community feedback process that feeds into changing and updating our data products over time. So I talked a little bit about advisory groups, just to tell you more about them. If anybody is ever interested, we are always looking for people to join our technical working groups, and those opportunities are usually advertised in August around the time of the ESA meeting. We have over 20 technical working groups currently. They're composed of experts in the field, and we try to make those groups a mix of career stages. Grad students, if they have the time, are awesome on these twigs, all the way up to people in mid to late career stages. And we try to get folks from agencies, USGS, State Department of Natural Resources, and also from research institutions and universities. So technical working groups can be general or can be set up to answer a specific question. So for example, I have run 2 different technical working groups. Right now, we have a general aquatic biology technical working group that covers everything aquatic biology, and we just have this wide range of expertise that I can go to and ask questions. In the past, I've set up more specific things like an algae technical working group. Like I mentioned before, when data products and things change, we go through this internal NEON review through the integrated product teams, which are a mix of scientists and managers from different teams across NEON. And then we have the STEAC, which is the Science, Technology, and Education Advisory Committee. And that, again, is a mix of external scientists that work on how NEON can meet its high-level vision, talk about science requirements, strategic engagement, and prioritization of activities to fit within our budget. But ultimately, NEON is about data. Data need to be discoverable and actionable by a broad audience. Like, if people can't find our data, again, there's no point. They need to be able to find it. Our portal needs to be clear and easy to use. It needs to be intuitive. We provide tools such as workshops, tutorials, and R code to help users. And then as we get further along into the 30 years, the data will be bigger and bigger and bigger. And so we need to also provide the tools for people to work with those bigger data. And so NEON's data portal is set up sort of as a three-pronged approach. We have information on context, engagement, and data. So the context, if you go to neonscience.org, history, a lot of the stuff I've talked about here, information on data and samples, field sites, you can explore the field sites, see pictures, create your own maps if you want of those sites, and then some impact about publications. We also have the observatory blog and the newsroom, which you can sign up for more information there. And there's a ton of other stuff. This is just sort of a smattering of what is on neonscience.org. We also have a lot of engagement resources there, such as workshops and courses that have been recorded and can be watched again, code-alongs, code hubs, our user-submitted code. So if a researcher uses NEON data, comes up with some pretty cool code and wants to share it, they can share that via our code hub. We have the Learning Hub and tutorials. So often those are workshops that we have done before, or some are very simple, like here's how you download and access NEON data using R. And then we have that research support services group that I talked about before. And then ways of getting involved, collaboration opportunities, advisory groups like I just talked about, and career opportunities. And then moving into the data portal, this is at data.neonscience.org. You can access all of this. You can go back and forth between the two websites, no problem. And these data can get pretty big depending on what data products you want to look at. Again, we have over 180 different data products to look at on the site. And we have time series data for airborne observations, terrestrial instrumentation, aquatic instrumentation, and then terrestrial and aquatic observations. So those are the samples that are collected, samples and data that are collected by our field ecologists. All of the data product landing pages also have links to data documentation. So we provide our field protocols, lab SOPs. Often for many of the data products, we use an external expert lab to measure chlorophyll or identify bugs. We have data quick start guides and data availability, and all of that is available on the data product landing pages. Tons and tons of information. But what's super, super important for us to do is to ensure that we are providing good quality data. We do that through a combination of quality assurance and quality control. So we want to make sure that sensors are correctly installed. They're installed at the right height, facing the right direction. There's a whole suite of things that go into sensor installation. That values are coming in within the expected range. Nothing is crazy high or crazy low. We want to make sure our data are free of typographical errors and that labs and technicians are using the protocols and the SOPs correctly. And then we have quality control where we look at unexpected gaps in data, especially with sensors. They sometimes are able to do some gap filling and fill in those gaps if there was an outage or something. We look at values that are outside of the possible range, processing errors, and publication errors, such as data being in the correct format once it gets to our portal. And we do data quality at every level throughout data collection and the observatory. So this starts in the field where we train on standardized protocols. We collect data via apps, and that allows us to have more control over the data that are coming in. So we can control ranges of data. We can have certain fields that pop up that are the only ones that are allowed to be used. We can have validation rules like this number has to be lower than this number. We provide regular office hours between our field scientists and our headquarters science team so we can chat, and if there are any questions that come up, we can just talk about them in person. We have a problem reporting system. And then we have field audits that are performed by a traveling Battelle audit team, and these are incredibly valuable. That's where one or two people go out, watch our field scientists either calibrate instruments or collect samples and see how well they're following the protocol or if they've started to deviate over time, which happens sometimes. And then for our data ingest, we have a variety of validation rules. So we look at ranges, accepted values or constrained values, things like that. Again, we don't want any numbers that are so high that they don't make sense. We use taxonomy tables to control spelling and references and things when we are looking at taxonomy data. It's really easy to spell scientific names wrong, and that can really affect the data. We have controlled lists of values, and then parser 2 creates, where sometimes data will come into our system and we can have some calculations that create a new field without having people in the field try to do those calculations in their head or on their tablet or something. So those are ways of making the data quality more consistent. We do regular data checks as well. We have monthly and annual R code that runs via R Markdown and produces reports. And so we on the science team are looking at those every month, and dealing with any errors or missing data or plausibility that comes up from that. We have an annual data release that happens in January each year, where we look at released versus provisional data. And then we have a calibration validation team that performs audits for contracting labs, calibrates sensors on a regular schedule, and then sensors rotate among the sites over time. And then if you go to look at our data, if anything has happened, we have found errors, we've had to make changes, we have issue logs on all of the data product landing pages that can tell you what happened, why, how the data were fixed, how you can use the data, and that's really important to look at. And then we have NEON data notifications, which you can sign up for at neonscience.org, and they'll come right to your email if something big has happened. In addition to this, we optimize our data. So we want to make sure that we are providing the best data for the community, and we're providing what the community actually wanted. So as time goes on, it's important for us to take another look at our protocols and our sensors and things like that to see if the data we're providing are getting used, if they're good quality, if they make sense, and if they're what people want. So we do this in conjunction with our technical working groups again. And we use this to sort of optimize our design, solve problems that come up. And this is done through a process of using statistics on our existing NEON data. Now that we have several years of data that we can look at, we're able to perform these analyses and see how well the observatory is performing. So how do we know what we're going to optimize and when? Well, there are some that are planned analyses that are prioritized based on sampling effort and likelihood of success. For example, there was a optimization that was done on root sampling at the tower plots that said that our field ecologists could collect less and it wouldn't affect the data quality, which is great. So a little reduction in staff resources. And then there are things that are unplanned, like something that I worked on recently. And this is a challenge faced by our field staff, where our staff that samples in Blue River in Oklahoma experience very hot conditions during the midsummer, so hot that they can't be outside all day long. And they weren't able to get all of their work done for aquatic plant sampling in one day, which is important because if there's a thunderstorm or something and the water rises, the next day is totally different conditions. So we need that work done in one day. Through optimization analysis and using statistics, I was able to determine that we could decrease the number of points collected by half. They could collect everything in a day and it didn't affect data quality, and they could get out of the field before it got way too hot for them. So to date, there have been 64 optimization analyses completed, and that spans 42 different data products. So we've really started to look at a lot of our data. There are 9 optimization analyses currently in progress for 2025. And I was going to show you a quick example. I'll run through this really quick since I'm sort of running out of time, but this is a big optimization that I have been working on since 2020 and is still going because it's big. So for our algal taxonomy sampling, we have this whole suite of instrumentation, biogeochemistry collections in biology that we collect for algae in our streams, lakes, and river sites. I'm talking about algae today. We collect algae at all 3 of those site types, streams, lakes, and rivers, and we collect benthic algae, so growing on the surface, the bottom of the lake or the river or the stream, and then we collect phytoplankton in the water column. And we started collecting these data in 2014. We collect 3 times a year, roughly spring, summer, and fall. We collect a sample in the field, and then that sample goes back to our lab, our domain support facility, where our field ecologists subsample for taxonomy, and then they get some chemistry subsamples where they can look at chlorophyll and things like that. What we found out after starting to work with more data in 2020, one of our expert taxonomy labs that we were contracting with called us up and said, hey, my data looked different from the previous lab. what's going on with that. And so this is actually a really common issue with diatoms and with microalgae in particular, because the smaller something gets, the harder it is to ID consistently, right? And so basically what you can see here is that green is one lab, red is one lab, and blue is one lab. And even though they were looking at data from the same sites, you can see some clustering by lab. So that means that there's a lab or a taxonomist effect, which again is very common with diatoms, which is what these are. So what do we do with this? So this compromises the data user's ability to evaluate spatial and temporal data, right? Because they're going to come in, look at our data and say, oh, well, in 2019, this community totally changed, but really the lab changed. NEON's a 30-year project, so we need to figure out how to deal with this before the data issues compound over time. So what we did was got community input. I set up an algal technical working group back in 2020, and this was specifically to answer this question. And this was a group of like 12 experts in the field who could help us come up with a new plan. After that, we came up with a plan. We took that to the IPTs, the integrated product teams within NEON, and from there it went to STIAC, and then a memo to the NSF. And in there, we also were able to do a community survey to poll anybody else in the algal community and see what data they actually wanted provided. We were able to do a pilot study to assess the lab methods we were using. And now we are in our final stage, which is our last pilot study. We should have data back by the end of the month, thank goodness. And then we'll be able to move on with changing our data product, which is really exciting. And basically what we were doing, instead of collecting 8 samples across the stream on a certain date, we're going to collect those 8 samples and then composite them to remove some of the variability. And then we are setting up a voucher collection at the external labs with photo images. So one lab can look at the other lab's images and be more consistent over time. I'm going to skip through a bunch of these slides and kind of get to the end. So this is the main thing that we needed to do to make this analysis work, and that is to add vouchers. And this has been published in by Sarah Spalding and other diatom researchers, and is starting to be used more and more in analyses. The EPA is developing a voucher flora so they can be more consistent across their regions for their monitoring programs. But it's not really in use for large spatial and temporal projects like NEON yet. So we're really trying to come up with this new plan for NEON, and we think that it'll probably be interesting and be used by other researchers as we go. So as our taxonomists look at an algae sample, they create vouchers and images as they encounter them, identify them, get some consensus with other taxonomists, and provide those images and plates that the next taxonomist can refer back to when they're identifying data. And then if somebody comes to use our data, they'll know what set of images that taxonomy came from, and they'll be able to reference back to that. So that'll be a much better long-term reference of how the data were collected and how they were identified over time, instead of just wondering what taxonomist A called this and what taxonomist B called this. Again, I'm going to skip through towards the end. So this algal optimization that, again, has been in progress since 2020, it took a ton of people across our algal technical working groups, our aquatic biology technical working groups, some external labs, and our NEON data science folks. So you can kind of see like what all goes into this. And again, that's not even thinking about the fact that this went through our STIAC, through a STIAC review into the NSF for concurrence. So there's a lot that goes into changing and updating and making these data products better. So what's new with NEON now? NEON is kind of embarking on its 5-year strategic plan, where the goals are to enable science through partnerships, maximize operational excellence through the availability of high quality data. So I've talked a lot about data quality already. Harness evolving technology to catalyze discovery, enhance discoverability. Again, if people can't find our data, they can't use our data. Make the data more accessible and more usable and more clear and intuitive for people to use. And then foster an engaged workforce. It's really important to us to keep our scientists and field ecologists around because they know a lot of stuff, and it's hard to train up new people all the time. Um, so I just want to talk a little bit about the partnerships. There's a lot going on now with partnerships with other groups like the Ecological Forecasting Initiative, the Imageomics Institute, and then there are several international partnerships. There are other long-term ecosystem observatories being set up across the world. So European long-term ecosystem research, there's us, there's groups in Australia, South Africa, Finland, China, and then there's a new partnership with the South Korea group. I'm going to skip through these again. I think we've talked mostly about this. But in conclusion today, I just wanted to reiterate that the NEON Observatory was designed by the scientific community. Data products were initially selected by some of these workshops that happened back in the early 2000s. And sampling methods are standardized and designed really by the data users for the data users. Everything that NEON scientists come up with have to be vetted and go through our data user community to make sure that's A, what people want, and B, it's something that's going to work and provide high quality data. We always want to maintain high quality documentation and data on our website, so please do check out neonscience.org. And we provide data analysis tools for big data. So a lot of R code, there's some Python packages and things like that that are starting to come out to work with the NEON data as they get bigger and bigger over this 30-year time span. And then we're continuously optimizing and looking at our data products and the data that we're providing using statistics and community input. Even though this is a long-term study, it's 30 years, we don't want to like fully change course in a data product, but there are enhancements that can be made, and there's technology that we need to keep up with over time. So those are decisions that definitely go into that process. Are we still producing the same stream of data over time? Are we enhancing this? Are we doing this a little bit better? And then we're partnering with other networks, which is really great because we can't be an observatory on our own. We need to be working with other researchers and other networks across the world. So we're just looking at North America, and if we can be more interoperable with groups that are working in Europe or Asia or Australia, that just really increases the power of what we're doing. So with that, I'd love to take any questions.
Well, thank you very much, Stephanie. That was a fantastic presentation. The size and the scope of the work you guys are doing over at NEON is kind of mind-boggling. We have a few questions already in the chat. Let me see here. My chat just disappeared. There, it's back. The first question is, and I think you partially answered this. Do you communicate, coordinate, or try to harmonize with similar long-term projects elsewhere, and more specifically in Africa? And I know you mentioned the work with South Africa, but if you want to expand on that anymore.
Yes, that is one of the international partnerships that's going on. So specifically with Africa, it is the South Africa Environmental Observation Network. And NEON has been partnering with them through the Global Ecosystem Research Infrastructure, or GERI, group. And so that is a group of international observatories really that are partnering together. And I don't know the details of everything that's going on there or how much the data are harmonized. I know a lot of these, I do aquatics, and I know a lot of these groups are more terrestrial based. So I don't know the specifics of the data they're producing.
All right. The next question is, what aspects of the NEON program do you think could be most applicable to projects just getting underway in resource-constrained environments?
And resource-constrained meaning like funding?
Yes.
Or, okay. I think leveraging the community is huge. We have found through NEON that a lot of people want to be involved and want to be on those technical working groups and want to help us out. So the more that you can get the community involved and into your network and providing that technical expertise and suggestions on on how to sample and how to do the work, that's great. And then on top of that, like it's just hard to get, I don't know where you would want to be working, but it's hard to get around. So if you can find people in those areas already that could do the work for you, you wouldn't have to be traveling around to collect samples or things like that. Um, I don't know if there's any thoughts to using citizen science. We don't do that too much with Neon, although there were some talks about doing it early on. But yeah,
And
I think leveraging the community as much as possible. Um,
I'm gonna kind of selfishly ask a question on top of
yeah.
that. What about like, so I think, watching your talk, there's like tons of stuff. Obviously, there's all the protocols. So like, or how portable do you think that those protocols are for sample collection? And then I think as someone who's worked on a database in the past, getting an operational database that can handle that amount of data, the different types, the amount of data is a huge effort.
Mhm.
Is that something that Neon is willing to share, or at least provide advice on setting up a database for environmental monitoring?
Yeah, so for the first question, how portable? Um, Speaking for aquatics, our protocols are really portable. Um, I know they're mostly based on existing work, existing protocols that people do. And so I've heard of people taking, for example, the aquatic macroinvertebrate protocol that I've written, and they're using it in their own streams just so they can follow our protocols and do their own work in their own lab, right? Some things are less portable. Instrumentation is a lot tougher, obviously. And I would think a lot of the terrestrial work would be pretty easy to do in multiple places as well. I know some of the tougher things for them revolve around like small mammal sampling and things that you need more permits for. But for aquatics, like no one really cares if you collect bugs. So they care if you collect fish though. So permitting is tougher. Um, and then the second question about the database, yeah, that is Neon built its own database, right? And that's a lot of work, and there's a whole group that did it. And I think if you were to reach out, if your group reached out to Neon, I believe those people would be happy to talk to you because they're always going to conferences and putting together technical working groups and things like that, trying to make everything more interoperable. And the idea is like the more interoperable everything is, we can see your data, you can see ours. And I think there would be definitely some things that could happen there.
Very cool. Let's see. The next question is, sorry if I missed it, but how tall are your towers and biotics and the biotic samples they collect, and or what biotic samples do they collect?
Yeah, you didn't miss it. I didn't talk about that. So towers are designed to be above the forest canopy. So I don't know the exact dimensions. Again, I'm an aquatics person, but they are some distance above the top of the canopy. So I know the tallest tower is in Washington State because that's the tallest forest canopy. And then like our towers here in Colorado on the plains, there's no trees, and those are about a story tall, like a building story. They're pretty short. They don't collect biotic samples. They just have instrumentation on them that collect atmospheric measurements.
The next question is, are any of these habitats in more disturbed areas or on the wildland urban interface? And if not, is there any plan to integrate some of these sites into habitats like that in the future?
Yeah, that's
Habitats
a
on the
great.
woodland. Oh,
Oh,
sorry.
sorry. Go
Go
ahead.
ahead. I was
No.
just rest of the question. Habitats on the woodland urban interface would be of interest for many groups, including mosquito control
Yeah.
applications.
Yeah, that's a great question. Neon was initially planning to do more of that. Turns out, some of that's really hard to get permitted because it ends up being like in people's yards and things. But um, so we have those core sites that are thought to be like less disturbed, more representative of the region as a whole. And then we have these gradient sites that tend to represent, well, more of a gradient, right? Like they could be closer to that interface. I can't think of any that are directly like within a wildland urban interface. Um, Initially during site selection, we were trying to have exactly a site like that, actually near Bozeman, Montana, that was in a suburban area, and we just couldn't get it permitted, couldn't do the work. So it depends, like, because we have such permanent infrastructure there.
Mhm.
So that's a big deal. If you were kind of like you were saying, Justin, having more portable methods and kind of running in and collecting samples, that seems like that would be easier to do in places like that. As far as adding sites, I don't believe we are planning to add any sites right now because it's a big ask, and we have the budget that we have right now. Um, but things like the mobile deployment platform can be requested from Neon, and you could put those in any of those places if you wanted, and that would be a great way to kind of use the existing Neon infrastructure, stay within the standardized methods and data collection, and kind of look at your own questions.
Very cool. And I guess maybe a follow-up question on that is for most of your, and maybe you mentioned this and I missed it, are most of your collection sites, are they on federal land? Are they on privately owned land?
It's a mix.
It's a mix.
Some we have some Forest Service land. We have some that are like Nature Conservancy land. We have a couple sites at national parks. Um, and then some that were existing research facilities, um, places where LTERs are happening, things like that. So
Right.
it's quite a mix.
Quite a mix.
And we have a permitting
Um,
group who deals with that.
oh, yeah, I imagine. It's pretty complicated work.
Yeah.
Um, the next question is, Stephanie, thank you for the wonderful presentation. Can you say a bit more about the samples in Arizona? What kinds of samples? Are there any space considerations? How long will they be stored? Again, congratulations.
Thanks. Yeah, so Arizona State University houses the Neon Biorepository. They have their own website and portal you can go check out. It's like biorepo.neonscience.org, I think. Um, they So initially in the planning, we planned out the number of samples we thought we would be sending them, what size containers everything would be in, how everything would be stored. Are things stored? There are things there that are stored at like minus 80 degrees. There are things that are ambient. There are things that are preserved. There are things that are dried. So they have a whole range of things there. And then they have curators who basically work in the museum. Some like specifically work on plants, some specifically work on the macroinvertebrates or the mammals, and they kind of split it up that way. So there are space considerations. However, they built a facility for Neon based on that initial um, plan that we put together, which again had to have all the bottle sizes and everything we thought we'd be sending them. So anytime something changes or we come up with something new that we think we want to send them, which does happen sometimes because it's tough to throw things away. If you have a sample and you're like, we should probably just save that. Someone might want it someday, you know? Making sure they have the space to store that is really important. And sometimes they don't, and that is a consideration. But they are planning to store neon samples for the life of the neon project, at least. I don't know where it goes from there. So that's 30 years.
And I had a follow-up question based off the samples. So you mentioned that some of the samples can be requested for sequencing. Are any of the samples sequenced right off the bat?
Yep,
Or
we sequence. We do aquatic and soil microbe sequencing. I believe they do beetle sequencing. I think there's some mosquito sequencing as well. And then there's some fish barcoding too, just to check species. And then we also do aquatic macroinvertebrate and zooplankton metabarcoding, so that's more of a community sequencing analysis. They get them all. But yeah, there are a lot of data streams that are already on our portal that are already sequenced. And then anything that sequenced the DNA extract goes to Arizona State for archiving if people want to do more with it.
And do you guys house that on your own database of sequence information? Yeah,
Yes. Yep.
very cool. Let's see. We just had another question come in, and I know I had a lot of questions after watching you talk, so I have one after this. But so Neon had about 10 years of planning. Are there key lessons from this planning phase that we might use if we wanted to establish something similar, but on a shorter time frame?
Mhm. Yes. like I mentioned before, I think that community involvement is really, really important. I think it also depends on like where your funding's coming from and how they want to handle it, right? Like NSF had a lot of oversight over what Neon was doing and kind of the last like final review and is this meeting the needs of the grant that we're providing you, right? So working with your funding organization and working with the community. And then if you can leverage things that are existing, like if you could use something from Neon or one of those other global networks, I find that the easiest when I'm working on protocols and sampling. Like if I can leverage things that USGS and EPA are already doing, that is the best thing because we know those are tried and true ways to collect data. So if you can find existing projects that are doing that work, maybe longer term projects or common practices in the field, I think that would cut down. So we had to come up with a lot of this from scratch and put it all together, but hopefully not everybody else will have to.
I imagine there's quite a few lessons that could be learned.
Yeah.
My former PI used to always say, never reinvent the wheel.
Yeah.
It sounds like you guys have built quite a few wheels. Um, Speaking of your data repository, you mentioned integrating new tools, and of course, AI is big in the news. Have you guys had any thoughts about integrating AI into your data set? Because I
Yeah,
imagine
there
like
is a
400
lot of talk
terabytes a year.
about integrating AI. I don't know the specifics of it, but I know there are several conferences that people are going to shortly to talk about AI, and there, I believe, is a whole AI workshop coming up in June or July that Neon's participating in. So yeah, so for now, I know one of the big AI projects has been with I believe it was in Hawaii with bird calls and audio data. And there's a big AI project, and I don't have the name of that offhand. Um, but I think it was part of the Imagiomics Institute. Um, and there are other AI tools that I know are being thought about and ways to analyze the data and get more data really using AI. So don't know the specifics, but there will be more coming on that for sure.
Very cool. All right. I don't see any more questions in the chat. I'm going to go ahead and I'm going to ask one more, and then I'll close this out. What are some of the biggest challenges in maintaining data quality across such a large and diverse set of field sites?
Um,
I
yeah,
imagine
this
there's quite a few challenges.
it's a lot of challenges. I mean, this is the majority of what I do now is work on data quality. So different data streams are different. So people who work on instrument data have different challenges than like I do looking at observational data. But just figuring out, like, first of all, having enough data sort of in the history to be able to compare it to. So we're at the point now where we have at least 5 years of data for most of our sites. So we can start to create some ranges and things like that. And we can see if something's like far outside the range or if something's plausible. Just even finding the problems, coming up with the tools to find problems with data quality. We've been working on we do this through our code, our markdown, um, trying to figure out what set of standard tests we want to always look at. Like we want to look for, are all the data that we're expecting there or some missing? That's the first one. Are things within the right range? For taxonomy, are the taxonomy plausible in that region? And that can be really hard. It's easier with some taxa, but like macroinvertebrates and algae, everything's everywhere. So that's hard to say. And then just having the time to like look at those reports monthly and yeah, just having the time to review those and see where the problems lie. And for me, at least with the observational data, we also use external contractors. And so that's another variable in the process. So we are having, a lab identify or count the zooplankton and or measure volumes or measure our chlorophyll. And are they doing a good job? And do they have their own data quality practices that we can rely on that are working? So just knowing that everything's working and spending the time to have people like have eyes on the data, eyes on some of the processes, I think is probably the hardest thing. But super important. And then the decisions, like what do you do with something if it's seems off.
Right.
So,
Well, yeah, with 400 terabytes a year, I imagine
yeah.
that's an extensive
Yeah.
endeavor. All right. Well, I'll go ahead and close this out. I just want to say thank you so much again, Stephanie, for that fantastic presentation. And thank you to all of you for joining us today and contributing to the discussion. We hope you'll join us again next week on Tuesday, next week, not Wednesday, at 11 a.m. Eastern. For our fourth session in the series, we will be joined by Dr. Damaris Matoke and Dr. Eric Kokomo, who will be presenting on malaria vector surveillance for native and invasive Anopheles species in Kenya. Once again, we invite you to visit the refreshed Gene Convene Virtual Institute, where you'll find new resources, tools, and publications. While you're there, please consider subscribing to our newsletter to stay informed on upcoming events and activities. Thanks again, and we look forward to seeing you next time.